Hello, and welcome to Lecture 7, the last lecture before the midterm. We're going to finish up talking about postsynaptic potentials today, and then talk about synaptic integration. We're going to bring to bear our knowledge of basic neurobiology in considering the nervous system activation of muscle, in particular, how the neuromuscular junction gets activated. And then talk about going to our new unit, which really is the rest of the course, talking about the neural organization of the motor system, and talking today mostly about anatomy in the spinal cord and brain. Okay, so first, some discussion of neurotransmitters. Okay, so as I mentioned last week, we talked about synaptic transmission, and neurotransmitters are the molecules that are exocytosed by the presynaptic terminal that float to the synaptic cleft and bind to receptors on the postsynaptic neuron, which can then affect membrane potential. Okay, now the process we talked about last week involved what we call ionotropic receptors. Those are receptors which mediate a change in ionic permeability to channel directly. So what that means is that the ligand binds to the receptor, and that causes changes in permeability in that same receptor. These responses can be very fast, to the millisecond range. I just wanted to point out there's another kind of receptor, what we refer to as metabotropic receptor, an example of which is the G protein coupled receptor. In this case, we again have a change in permeability due to the neurotransmitter binding to a receptor, but it's much less direct. It's mediated through G proteins and sometimes through secondary messenger processes. And so what it does is it causes permeability changes, but in proteins that are somewhat distant from the actual receptor. Okay, so notice here that where the ligand binds to the receptor is in a different place than the channel which actually mediates the permeability change and causes the postsynaptic potential. What's nice about this is that what's not so nice about it is that this can take a lot longer for it to occur, though it can be much longer lasting, and also it can cause a much wider spread change in permeability due to a signaling cascade that ultimately, again, originates in this G protein complex. Anyway, I just wanted to point that out, but for the rest of today, we're going to be talking most about these ionotropic receptors. Okay, so here's some examples of neurotransmitters. These are probably four of the most common neurotransmitters in the nervous system. There's two excitatory ones, acetylcholine and glutamate. Glutamate is probably the most common one, a common excitatory neurotransmitter in the vertebrate nervous system. Acetylcholine, though, is the one that is used at the neuromuscular junction that we'll talk about a little bit later today. These two, they exert their effects by changing the permeabilities of sodium and potassium, as you'll see. There's two major inhibitory neurotransmitters, GABA and glycine. These mediate their effects by increasing permeability to chloride, often using this shunting inhibition mechanism that we talked about last week. Now, one thing to point out here is that, because it's saying here that the major locations are interneurons. What are interneurons? Interneurons are neurons which project locally. They connect up neurons with other neurons. The neurons that project to neurons only in the nearby locations within a particular portion of the brain. Generally speaking, long-range connections going from one part of the brain to another are excitatory, whereas short-range connections can be excitatory or inhibitory. Okay. Now I should point out there's many other neurotransmitters: dopamine, serotonin, norepinephrine, etc. These are ones you'd learn about more if you took the Intro to Neurobiology class. Dopamine is one that we'll get back to in a few weeks because a deficiency in dopamine plays a role in causing Parkinson's disease. And we'll talk about that later. Okay, now what I'm going to mention, we're going to go into into more detail later is the role of acetylcholine at the neuromuscular junction. Okay, but that's it for this part.